I'm very glad to be here online. Yeah, as you told that this year it will be not about goats. I know that you are always waiting for sweet goats, but this time it's not less sweet. Our research of sea urchin, thanking to Estefania Paredes and Erasmus Plus program. And the topic of my today's presentation is fertilization rate and larvae abnormalities rate of the sea urchin after the use of cryopreserved sperm. Keeping of laboratory animals that are very useful model objects for biology, biotechnological research, and for other research, because as you know, sea urchin, for example, were the model object while studying the fertilization and were the first who gave us a possibility to make in vitro fertilization both for animals and humans go further. So the problem is associated with the such things like seasonality of access of mature animals, because, for example, sea urchin are taken from the sea, from the ocean. Their genetic heterogeneity that can affect the results of scientific research, and sometimes the research might fail. So the cryopreservation of gametes, and in particular sperms, allow to preserve genetic material for a long time and use this material for your further breeding and for your laboratory research throughout the year. That's why our research aim was to evaluate the fertilization rate and the rate of abnormalities of larvae of sea urchin after the use of cryopreserved sperm. So we use the sperm that we obtain using the dry method. So we just crushed the sea urchin and aspirated the sperm that you can see on a photo, that the white liquid, and try to avoid the contact with water, because after the contact with water, the sperm is activated and it can live less if we activate the sperm. Immediately, we calculated the sperm concentration and sperm motility. And afterwards, we aliquoted the semen.
Anastasia, let me stop you a little bit because we cannot see the slides, just the first one. So if you don't mind to share.
Okay.
Now it's okay. Yeah, we are on meta section. Thank you.
If I now change slides, do you see them?
Yeah, everything is okay now. Thank you.
And I now switch to another one. Do you see this?
No,
slide?
still the same, beyond methods.
Okay, I will be like here, okay? So this is the photo where you can see how we obtain the sperm. You see the white liquid inside the sea urchin. So this is the semen that we took into the plastic tubes without contact with water. And on the photo that is down, you can see exactly the sperm that we calculated motility and concentration. We aliquoted semen and mixed with DMSO solution in filtered water. So the final concentration of DMSO was 15%. We equilibrated the semen at room temperature for 5 minutes, then filled the straws of 0.25 ml straws. Placed that straws 5 centimeters above the liquid nitrogen, kept for 8 minutes, and then plunged into the liquid nitrogen. We thawed the straws on a water bath at 35 degrees for 6 seconds. And then we washed the semen to get rid of the cryoprotective medium, gradually adding the filtered seawater, and we were observing how the motility was restored. When we saw that motility restored higher than 65%, we understand that we can stop. We obtained the fresh oocytes, the same method as we obtained the sperm, also by crushing the sea urchin and picking the oocytes from the inside. We mix the oocyte with sperm in ratio 10 sperm for 1 cell. And then after 1 hour of incubation in filtered seawater, we determined the fertilization rate. I don't know if you can properly see, but when the sea urchin oocyte is fertilized, the zona pellucida is going a little bit out of the oocyte. So we can understand, watching on that the oocyte is fertilized. On the second day of cultivation, we calculated the abnormal larvae rate. On a photo that is down, you can see the larvae. It looks a kind of Eiffel Tower for me. So we calculated which larvae are normal, which are abnormal, and then we made the abnormal larvae rate. And the control group was the group of sea urchin oocytes that were fertilized by the fresh semen. So what we found out in our research that cryopreserved sperm has significantly reduced fertilizing ability. As you can see, that fertilizing ability was almost 40% lower than using fresh sperm. However, 60% of fertilization rate is also fine. The larva abnormality rate, we had no significant difference between our cryopreserved sperm group and fresh sperm group. So we can say that cryopreserved sperm, cryopreservation of sperm at all, didn't influence so much on the proper development. However, the fertilization rate drops. And to conclude, we can say that after cryopreservation of sea urchin sperm, we receive lower fertilization rate, 40% lower. However, the abnormality rate of larvae is on the same level as after using fresh sperm. I'm very grateful for your attention. And I want to tell thanks for Institute of Cryobiology and Cryomedicine for this possibility for me and my colleagues from the Kharkiv National University to go to University of Vigo and to perform this amazing research. I'm very grateful for Stefania Paredes for her support during that 3 months of stay and now as well. And I'm thankful for Erasmus Plus, also for this great possibility. Thank you.
Thank you so much, Anastasia, for such an excellent report. And sea urchins are cute. It's true, you're absolutely right. So now we can go ahead with questions. So if anyone has questions, just turn on the button, raise the hand, and you can ask questions. So you can just unmute your microphone and ask any questions to Anastasia. So please don't stay silent because it's like the worst thing you can do. Oh, I see the first questions. Natalia, could you please ask your question? You need to turn on your microphone. It's off.
Sorry.
Thank you.
Thanks for the interesting report. In your presentation, Nastya, you described the procedure of freezing straws. Please describe the quantitative indicators of this procedure. This is cooling rate, temperature before the sample is plunged into the liquid nitrogen, please.
Research about, thank you for your question, yeah. The research about cryopreservation of sperm and all the procedure and protocol was taken from the last research of the group of Estefania Paredes and her team. So we just took the protocol that was already invented and that was already processed. So you want to know a little bit in details what we were doing during the protocol? Almost everything is described in our material and methods. So we just mixed the semen with 30% of DMSO 1-to-1 very gradually, putting drop by drop. So there was no kind of osmotic shock. The final concentration, that's why would be the 15% of DMSO. The equilibration at room temperature was only 5 minutes, but that time we used for filling the straws. And afterwards, the group of Estefania Paredes made the investigation that 8 minutes and 5 centimeters above the liquid nitrogen was the perfect time and the perfect distance from the liquid nitrogen. So the cooling rate was the best for motility and viability of spermatozoa. And afterwards, we just plunged the straws into liquid nitrogen and stored for how much time we needed.
Thanks, but I want to hear about temperature, sample temperature before the sample is plunged into liquid nitrogen. What temperature of sample?
It was
Can you say about it?
Yeah. From the literature, we know that it was about minus 120 degrees, these straws, because the research group was investigating that. But we know that only from the literature, we haven't performed this experiment ourselves.
Thanks.
Thank you so much for such detailed description of the protocol. So we have a question from Taisia YouTube.
Hello to everyone. Nice to see you, all of you, especially you, Nastya, because we miss you very much. And we also like your talk today. And my question was also about the protocol. I understand that you probably not you didn't change the main protocol, but probably you saw some information in literature, because 30% of DMSO is very high concentration, and it is toxic. So probably this affect the fertilization rate, and probably if you will change, you will low this concentration, it make it toxic. And my question about your knowledge in literature, have you found some information about another concentration or another cryoprotectant? Because usually for spermatozoa, glycerol is very popular cryopreservation agent. And another, my question is about, I know that you will go into test DNA fragmentation test, and have you probably some preliminary results about this? Thank you.
Thank you for your questions. So we have been performing, and the team at all was previously performing tests of different concentration of DMSO, and not only DMSO, but ethylene glycol as well, and glycerol as well. And after all that tests, we found out that 15%, however, it seems like it's a very high concentration, but preserved the best. Still, the time when we equilibrate is not so long. That's why, taking into the account the concentration and the equilibration time, this protocol worked the best for sea urchin spermatozoa. Still, I hope that we will have some more ability to test and to help Estefania Paredes and her team, maybe to perform more experiments. We will see. And regarding your second question about DNA fragmentation, we are still working on that, but we have done the viability test after equilibrating with DMSO and cryopreservation. And we had very interesting results. That's why we are not presenting it now, because it seems like DMSO must influence negatively the viability of sperm, but we had higher viability after equilibration. That's why we think that we will make more experiments, and if it really will be like that, it will be a shock, I think, for all researchers. But still, yeah, the viability was a little bit evaluated after 5 minutes of equilibration with DMSO, about 7% or like that, but it was statistically significant. So that was our shock. Let's see, our future research will show that. Maybe we don't know something about biology.
Okay, we'll hope.
Thank you so much. I see in the comments also that everyone are excited to hear about future developments and future steps. So that's why let's thank you, Anastasia, one more time, and let's move to our second speaker. That it will allow us to learn even more about sea urchins, but instead of the sperm cryopreservation, the focus is going to be on cryopreservation of larvae. And now I would like to call upon Alba Lago. So could you please share your screen